Good afternoon, ladies and gentlemen. Welcome to this evening's professorial lecture featuring Professor of Molecular Biology, Professor and President we're very sorry for the late start, but I think we'll catch up. Medical professors are not in the business of talking a lot, so I believe that that we'll do some catching up. I want to extend particular welcome to our distinguished Vice Chancellor, Sir Hilary Beckers. Also want to welcome the University Registrar, Dr. Maurice Smith, Principal Professor Denzel Williams, other members of the management of the UA Mona, deans, etc. I would like to also recognize family members of Professor Brown, particularly Mrs. Audrey Hawthorne Brown, his wife. Can you stand up and be recognized, please? Give her a hand, please. And of course, the children, Pearson, Princeton, and Aurelia. We want to welcome his schoolmates from Arden High School, and I'm so happy the deputy principal is not here this evening. Mr. Alok Jain, Professor Monica Parshad Asnani, and Charmaine Boris Keys. Give them a hand, please. We want to welcome Mr. Stephen Stewart and family from Wilbert Stewart Basic School. Mr. Edward McIntosh and Mrs. Audrey McIntosh, Mrs. Rose Thompson, Althea Anderson, family friends, and others from the Pentecostal Gospel Temple. Please welcome them. Colleagues, past student, mentees from the United States, Canada, Africa, Australia, India, Pakistan, and the entire world. In fact, we are streaming. And I see here we have colleagues from the University of Buffalo in New York. Please welcome them, Professor Diaz and Dr. Shakan. Also want to welcome former Principal Professor Dale Weber, who is here with us. Please give him a hand. Ladies and gentlemen, we're going to start with prayers. I'm not surprised that Professor Brown has invited Elder Dr. Frederick Thompson to pray. There is something that a microbiologist has in common with a man of faith. The microbiologists deal with things that are invisible, and people of faith, you know what faith is? The substance of things hoped for, the evidence of things not seen. Let us welcome Elder Thompson, please.
Thank you. Let us pray. Sovereign God, immortal, invisible, omnipresent, omniscient, omnipotent, we gather this afternoon in your presence for, Lord God, to attend to this particular effort that is being carried out at this time, the inaugural professorial lecture. We come, Lord God, to be edified. We come to be sensitized to issues and matters that has to do with our humanity. We pray your presence now, O God, to tabernacle with us. We ask, Father, that you will guide the process this afternoon, and even the one who will deliver this presentation. We ask that your Holy Spirit will now enfold him, embrace him, give him clarity of thoughts, clarity of mind, and the Lord God, the empowerment that will be so essential to bring across, Lord God, what is to be espoused here this afternoon. So now, God, we ask that your holy presence will continue with us, that you will give us, Lord, good retention and good recall, so that when we shall have left this place, the information that would have been garnered and that would have been imparted would, Lord God, be in us, with us, and we'll be able to use for the betterment of ourselves. So now, God, we ask your presence and your continued blessing as we give you thanks in Jesus' name. Amen.
Thank you very much, Elder. So, ladies and gentlemen, we are met here today as we continue our intellectual discourse, as we continue to position the University of the West Indies as a very important fount of knowledge. We generate knowledge here, and I think we have a responsibility to share it not only with the university community, but with the wider society. And we are very happy so to do. Of course, Prof. Brown professorial lectures are generally associated with newly minted professor. Prof. Brown has been a professor since 2017, but it's never too late for a shower of rain, because they say professors must profess. So we expect many more of our professors, however long they have been appointed, to come out and share the important research they have been doing with our community and with the nation. As you can see, the topic is genes and shots, Lepto and other stories through molecular lenses. The only thing I understand about that is the genes and the shots. But I'm sure by the time Prof. Brown is finished with us today, we'll all be edified. So, ladies and gentlemen, we have some preliminaries in terms of greetings. And we're going to have three greetings, one after another. Three remarks, welcome greetings. We're going to start with Professor Maxine Gossett-Williams, Deputy Dean. And we want to, in the same breath, apologize for the Dean of the Faculty of Medical Sciences, who is traveling. So Professor Gossett-Williams will give us welcome. She'll be followed immediately by our campus principal and Professor Tanisha Stevenson, in that order. Thank you very much.
Okay. Dr. Stanbury, can I get authorization to state all protocols observed in interest of time? There are so many professors in here. Vice Chancellor Beckles, it's really good to see you. Professor Denzel Williams, really good to see you as principal here today. And I'm really happy to see Professor Dale Weber. Really happy to see you and Mona. Many others, and I think one of the challenges with the virtual platforms that we need to figure out a way that we can actually see who is on. So I'm sure there are a lot of distinguished persons there that really we should recognize. So with that, yes, I'm here to represent the Dean. She really is sorry that she couldn't be here. She's one of the persons that, Paul, you've had a long relationship with. And I myself apologize if I keep calling you Paul. I think we've come together on this journey for a long time. So, Professor of Molecular Biology, I want to start with the tagline that you used to announce your plans today. All your friends, you sent out a WhatsApp message. Greetings. It's about time. I recall the arrival of COVID-19 to our shores. It was almost at the eve of the previous date that you had set for your professorial lecture. How you must have contemplated the pros and the cons of going ahead with an in-person event or doing the online pivot. You decided on postponement of your lecture, and therefore I want to reassure you that the time for this professorial lecture is right. At this time, I think we're all attuned to the high level of threat to human life that microorganisms pose. 27 million deaths estimated from COVID and still counting. Undoubtedly, the world now appreciates that the next pandemic requires a sincere commitment to collaboration and global coordination. That the needed discussions must relate to when the next pandemic occurs. Not if, when. We understand advancing our knowledge of microorganisms only gives us limited insight in their power to change human existence. The concept that good health relates to balancing of exposure to germs, we need to actually be exposed to some germs for good health. First, it's protecting us from the exposure, and it's constantly changing the algorithm and challenges the truthfulness of the old adage, what doesn't kill us makes you stronger. Prof. Brown, Paul, your contribution to understanding the power of microorganisms over human health is commendable. Yes, the timing of this lecture is right. It is important for us to remain vigilant and be continuously sensitized. And so I look forward to your sharing of your journey today. I am sure we will be totally entertained by speaking about jeans and shorts. Maybe 5-6 years ago, maybe before COVID, we never thought that talking about microorganisms would keep us, attuned. So I look forward to hearing you, Paul. Thank you.
Our distinguished Chairman Registrar Stanbury has done an excellent job by recognizing everyone, including those in the world. So I won't go over that, but just to say greetings to all nice and decent people and Professor Lindo. But ladies and gentlemen, let me take this opportunity to recognize our fearless and distinguished leader, Vice Chancellor Beckles. Let me also recognize our former principal, Professor Weber and Lady Weber, and also our registrar, our university registrar, Dr. Smith, leadership of the Mona campus, our deputy principal is somewhere online, registrar, deputy registrar, and all very distinguished ladies and gentlemen. Professor Carr, our former deputy principal, good to see you in these lecture theaters again. And also very distinguished ladies and gentlemen who have joined Professor Brown and also his family who are here this evening, and the goodly Reverend Dr. Frederick Thompson. This evening, let me firstly use the opportunity to offer heartiest congratulations to our distinguished lecturer this evening, Professor Brown. As registrar indicated, you're talking about jeans and shorts and all the other things, but the only part that I'm interested in is to hear stories. And so stories are important things to help us to learn. So I'm looking forward to what the stories are. But this evening, ladies and gentlemen, it is not just about a professorial lecture that Paul will be delivering, but it really speaks to a broader context as to why we're here as an academic institution. This professorial lecture falls within the ambit of thought leadership. And so, as an academic institution of very high repute, it is very clear to me that what distinguishes us from all the other places that do offer certification is exactly what Professor Brown is about to do this evening. What he's about to do is to show you why, indeed, the UWI is not only considered a university, but is considered one of the finest universities in the world, in the top 3% of the best universities globally. And that has come from exactly the kind of human resource that we have that is manifested in Professor Brown this evening. Because it's the quality of our research that really makes the difference when you're talking about what makes a university from what makes other institutions that offer degrees. And so I'm very proud and pleased that the Mona campus was able to be associated with somebody of the ilk of Paul Brown, who has taken the very painstaking task of sitting in the lab, spending sleepless nights, spending hours and hours of trawling through data and looking at all kinds of literature and identifying critical considerations to deal with problems that we now have today and problems that we don't even think about as yet. That's the beauty of research. And what it does, it provides us with a kind of toolkit that says to us, we are not here just to generate a set of graduates that are dealing with contemporary problems, but we're here to provide our graduates with an analytical toolkit that is helping them to solve problems that we have not even seen as yet. And so our professorial lecture this evening is really taking you to another level to show you that UWI is not just here trying to certify people for what we know today, but we are here to provide people who are going to shape the future of humanity. And so this evening I would be really happy to look and see all the things that Professor Brown, through his genes and shorts, Lepto and other stories, through molecular lens, will be unveiling to us. But I'm very confident that the work that is being presented or that will be presented this evening will not only find themselves in journals and periodicals, but they will find themselves in policy circles. Because indeed, a big part of our strategic plan as a university is a pillar that is called alignment. And alignment basically says we are taking the knowledge that we have produced within the academy and we're using that knowledge to link with industry so that we can find solution to our problems. Paul, I want to congratulate you this evening for taking the time to do the work, and we look forward to hearing the stories. Thank you.
Our Chair, Dr. Donovan Stanbury, Vice Chancellor Sir Hilary Beckles, Professor Denzel Williams, Principal and Pro-Vice Chancellor of the UWI Mona campus, and our former principal, distinguished Professor, Professor Paul Brown. Esteemed ladies and gentlemen, good evening. Good evening. You know you're a big deal when the executive management turns up at your professorial lecture. Or it could mean the power of the union, the power of the union. Ladies and gentlemen, I'm tempted to start with a disclaimer that the views expressed are those of the Deputy Dean of the Faculty of Science and Technology. And if well articulated, they are representative of the Dean of the Faculty of Science and Technology and the entire faculty. If not, they are the sole responsibility of the presenter. However, ladies and gentlemen, such provisos should not be necessary in this greeting, so here goes. On behalf of Professor Michael Taylor, Dean of the Faculty of Science and Technology, who is overseas and could not attend, and the faculty fraternity, we are pleased to bring salutations at the professorial lecture of Professor Paul Brown. Our faculty considers Paul Brown a very close affiliate of the faculty, and dare we say, even family. His foundations in microbiology in the Department of Basic Medical Sciences in Biochemistry and his professorship in microbiology has FST written all over it. For all of you who are not following, that's the Faculty of Science and Technology. However, we are proponents of collaboration, and this intertwining of the faculties of medical sciences and the science and technology faculty is admirable and worthy of replication. Prof. Paul Brown has blazed the trail on significant research endeavors in medicine and the physical sciences. From our FST lens, his work, for example, in relation to the impacts of climate change on leptospirosis, or on the environmental factors that precipitate increased risks to leptospirosis, has helped to advance the understanding of the health implications of climate change and other environmental factors, even before it became fashionable to do so. His molecular work in relation to pathogen is widely recognized, and his ascension to the rank of professor in molecular biology is deserving and celebrated in FST as it is in FMS. The faculty applauds the accomplishment of Professor Brown and lauds his research and advocacy that has enhanced the reputation of this institution. We're happy to be the launch pad of this esteemed colleague and many others. The views expressed, ladies and gentlemen, the views expressed. And so to ensure I'm invited to represent our faculty at the next event, let me quickly extend once again my congratulations and our congratulations to Professor Brown and by extension, his collaborators on this accomplishment. And please accept our faculty's best wishes for the next frontiers you will conquer.
Who's a scientist can talk, eh? Ladies and gentlemen, permit me to do my second round of recognition. I hope my WIGAT friends don't think that this was deliberate. We're good friends with WIGAT. We'll meet on Thursday at another. So I want to invite the executive, I want to welcome the executive of WIGAT, who are here to support their president. Please give them a big hand, please. We also want to welcome Ms. Ruby, Prof. Brown's mother-in-law. And his sisters-in-law, Caroline and Benitia Hawthorne. Welcome. And we take what the principal said very seriously in terms of alignment and our research, the findings of our research finding themselves into policy. So we did invite the ministers of agriculture, health, and finance. Unfortunately, they are not here, so we apologize for them. Other exciting things happening elsewhere downtown, I hear. So, ladies and gentlemen, we are going to get into the lecture, but we want to just set the mood. And so we're going to invite now special music from Mr. Vincent Ricketts on the saxophone. Please welcome him.
Thank you.
I think Mr. Ricketts deserves a bigger hand. Give him another hand, please. Ladies and gentlemen, to introduce Professor Brown, we're going to ask another member of the Arden Syndicate. He's online, and he has a, I don't want to be so strong, Principal. I think he has a recorded message. Our Deputy Principal is traveling, but it was good enough to record a message to introduce Prof. Brown. And after the that recording, the next voice you will hear will be that of Professor Paul Brown. Thank you very much.
Good evening, colleagues and friends. First, let me apologize for not being there in person, as this week is the week of the Caribbean Public Health Agency Conference, and I'm off as part of the organizers for that event. Let me congratulate Professor Paul Brown and the Department of Basic Sciences for accepting the task to host this latest edition of professorial lectures. Indeed, in the academic journey, the pinnacle is attaining the rank of professor, which signals to peers, students, that the individual has contributed significantly to their field of study. The UWI system for promotion to professor attaches weight and significance not only to scholarship, but to the contribution to the sustainability of the area of study by the awardee, being a teacher, mentor, and in fact, a student of that particular area of studies as well. In fact, the training of the next generation of scholar is perhaps, in my mind, the single most important attribute of being a professor. Professor Brown will be sharing with us a synopsis of his academic journey, where he will describe that he had a solid foundation as an Arden alum, which was then molded through his doctoral studies. Of note, you will recognize that the previous professorial lecture was also given by an Arden alum. He will, I'm sure, share with you the technical lenses that he uses to gain a better understanding of leptospirosis, an endemic illness within Jamaica. I now refer to Professor Brown as a microbiome man, and hopefully he will share some insights here as well, as it is that interaction between humans, microbiology, health and climate, which is the next scientific frontier. Indeed, I posit that if the world is to accomplish a sustainable development goal, as enunciated by the United Nations, that a better understanding of this interaction is needed. I'm therefore pleased that the Department of Basic Medical Sciences is leading the university's response in this area in a time of global flux, geopolitical tensions, and economic challenges, and at a time when many in the society seek to question the relevance of the University of the West Indies. Therefore, the hosting of professorial lecture events like these help to champion our scholarship, advocacy, and public educations. Congratulations, Prof. Brown, and I look forward to another successful professorial talk.
Okay. Good evening, everyone. So first, I want to go with what has already been established in terms of the protocol. But it is really good for us to be here. And it has been a while since I've contemplated giving this lecture. It was actually completed and ready for presentation in 2020. And then it was put off, and I wondered whether it actually made sense to do anything again. And the longer it stayed, is the more I was of the view that perhaps I shouldn't bother. But then there were several persons who kept prodding me. They are interested in this jeans and shorts business, and I had piqued their appetite, and they were thinking that I should actually do this thing. So here we are. First, I'm reminded of the dual use of information. Information that could be misapplied and pose a threat to public health, animal or environmental or natural, national security. But given that I didn't ask you to sign a non-disclosure agreement, I suppose anything that I say or that you have already heard can and will be used against me. So that is on me. But I ask you really to go easy with me, please. Thanks. The other aspect that I need to also bear in mind is the fact that in a lecture like this, there are different persons that are in the room and online. There are those that are generalists, those that don't care about the details that are in the particular studies, but they just want things that are of general interest, and I have to bear those in mind. There are those that are the specialists who I must deliver the details, or else they will never forgive me, because they need to learn something more than what they already know in the particular area. And then there are the others who will either flip-flop from one to the other, who are here, and thankfully some persons might not physically be here, who will be listening with one ear or looking with one eye, or hoping to see me sweat. And I did carry my prop just in case, because I need to be able to satisfy all of the persons that are here, to some extent, Registrar. So I won't define what are hybrids and chimeras and mosaic, but if you know anything about what you see around you, you will appreciate that a variegated leaf is variegated because of differences that are there, and hybrids are straightforward. And I could go on. But that is not the principal thing that I want to talk about. So there are 4 F's that are important to me. First is family. And I know that a picture like this will bring back memories for many persons who were born in that era. For those who are younger now, who are millennials or Gen Zs, they have no idea what Regal Plaza was before Azan is there and all of the things. But Olak Baby Show will bring back many memories of those things. And yes, I was born in 1968. This is my father, Percival Brown. He deceased in 1991 while I was in graduate school. My family at my wedding in 1997. I normally say that it is 20-something years, Audrey. 27 years hard labor of love. To the far left of the picture, that is the brother who was in front of me, Trevor. He's deceased. My cousin, first cousin, Len, also deceased. And then my mom. And on the right is my brother, Philip, and his wife. This is where we are now, and these have been introduced already. These are my the other brother, so I'm a wash belly. Last of the 4 boys for Miss Brown. She is 93 last year, so she's in her 94th year. I went to look for her today, and she is keeping on. He's still in the fight. So my eldest brother, Selbert, is on the far left, and then Philip, you would have seen him in the other picture of the wedding. This is Audrey and Miss Ruby, and her other two daughters, Venetia and Carolyn. I don't think they made it this evening, but they're probably online. Yeah. And this is Audrey's dad. Unfortunately, he passed away in 2018. So the two patriarchs have left us. And this is where we are now, give or take. And we're hoping for another 27 plus years. I know that some persons are well past that, right, VC? Okay, there's faith as well, faith in God. This is actually very important to me. In my first slide, you would have seen a THB, but you probably didn't identify it. I don't use it on campus, so I just list the academic qualifications. But I worship at the Pentecostal Gospel Temple on Windward Road, previously pastored, and I think most persons would be more familiar with Reverend Carmen Stewart, less so with the pastor who has been there now for 14 years, yes, Bishop Robert Stewart. I used to sing on the choir. For the dedication of the current church, I was singing first tenor, so I had a voice. But then I was playing football, and as a goalkeeper, which is in this picture, principal, this is at the MonaBowl, back in the day, then shouting at persons and singing were not compatible, and so one had to go. I was involved in Bible quiz, and I know my way around microphones, audio system, teaching, etc. Funding is another important F, and this is an important aspect of this slide because more grants doesn't mean more money. And if you're going to do good research, you actually need a good amount of money if you're going to actually satisfy what needs to be done. And so I'm actually happy for the NIH Fogarty grants that have been associated with, and thank our two collaborators from University of Buffalo. And, well, I suppose John was with, and still is with the other D43 that we have. But I want to use just the first one to typify how it is that things get started. So the first one is Jamaica Flour Mill Scholarship. So I got this in 1988. And this was to fund my final year at UWI after 2 years of student loan borrowing. And I want to single out, and he's not here, although he probably is online, a good friend, Dr. Neville Ying, who, if I remember correctly, was chairing the panel. And I convinced him that I wanted to make a contribution in biomedical sciences. I had dispensed with the idea, Monica, that I was going to do medicine. And although you cuss me dog rotten, I don't think I've regretted it. I think I am used to the handicap of not doing medicine based on how things have actually turned out. So I have 1 hour, thereabouts, but this quote has resonated with me. 1 crowded hour of glorious life is worth a lifetime without a name. And it is something that drives finding your place in the world and making a contribution because, in fact, you have very little time. And that time is really, and it is better to do all you can in the time that you have than, to hope that something will happen. You have to seize the moments. And persons who have helped me, and I want to single out 3 persons who have actually made this journey what it is. You can blame them for what I have become. Professor Errol Morrison refused to renew a one-year contract as a lab technician in biochemistry. Literally chased me out of the department. I will never forget that. I have forgiven him because he didn't know what he was doing. Or maybe he actually did know. And he thought that, in fact, it was the best thing. And we have had quite a number of discussions. But he thinks that I am creative enough and have potential enough to make it. And he encouraged me to go further than remaining in that position. Professor George Nicholson was the nephrologist at Cave Hill, Queen Elizabeth Hospital, former principal. The first time when I attended academic board was the announcement of his passing. And I couldn't, my intention at that time was really to come and listen to how big people talk in these meetings. And to hear that he had passed, and I was quite saddened. I knew that I had something to say because he had poured out all that he knew about the kidneys while I was studying in Barbados. And perhaps more than what the average medical student would know, I knew, and I had that as my disposal. And then Professor Eccleston Keane, who, even though Errol Morrison is a biochemist, I think that he is more the endocrinologist side, I would consider Eccleston Keane as the classic biochemist. And he taught me to love pathways, all that biochemistry is, and to the dismay of my students, because I require them to understand networks and gates and all of these things that go into not just pathways in biochemistry, but the fact that life is about pathways and decision making. And once you have entered through one pathway, then another one is closed. And it is difficult to go back because most of these processes are one way, except it is something that is in equilibrium. We are not living in equilibrium, based on how most of these go. All right, so the quick and dirty overview of genes and how they are inherited. So we think that we get half of our genes from our parents. That's half from the mother, half from the father. I mean, that is if the mother was Adam and the father was Eve, and then you are, But in reality, when you think about it, depending on where you are a product of several of these iterations over several generations. Then it begins to get interesting, because then it is not just a 50-50 contribution. It is actually much more complicated than that. And then it is harder now to trace where did you get these traits from. So, well, thankfully, none of my children have my nose, which is... And if I should go back to those persons who are sitting in that row on that side from Arden, and maybe it was an Arden thing that everybody from first form had names, and it was associated with something about them. Enough said. Principle, less is more, right? Less is more. Then there are mitochondrial DNA. So within the cells of eukaryotes or organisms, you have these powerhouses, and they have DNA in them, and these are inherited from the mother. So this is important if you're going to be doing studies that might involve identity tracing, for example, in forensic sciences. And then there is a Y chromosome DNA, because we have 23 pairs of chromosome. There are 22 autosomal, or those that are in the body. And then there is the sex chromosome. In this case, is the Y chromosome, and this is inherited from the father only. And then there are, for us, health factors. So you talk about the genes or the nature, what is there, and the environment, and then there are choices, which is a part of the nurture. And we'll come to talk a little bit more about this as we get into the rest of the talk. So, as a molecular biologist, I can't be afraid of who I am and where my DNA comes from. So, based on my DNA typing, I am 43.7% Nigerian. I am about 27% Sierra Leone, so West Africa. And then there is about 13% of my DNA that comes from Europe. I know from genealogy that I am getting my Africanness from my mother. And we can talk about that. And these were my parents at their wedding. This was 1957. They just came to Kingston. Right, so I've included this slide really to highlight what is possible in research. So back in the good old days, diabetes and hypertension research really was done in whole animals. And then we were able to do some work to include cellular and molecular studies. And I see at least one of my collaborators here, Professor Chooks. Thank you for being here. And the principal thing is that what we can do is limited by only our imagination. It's one of the first lessons. There is nothing that is really impossible if you really intend to do it. And even if you can't do it here, as I tell students, there is a collaborator, if you can find that person, where you can go and get it done. But it doesn't come easy, and I will share why this is so. My lab, I want to highlight the real heroes whose work I'm going to be presenting tonight. Stacey Stevenson Clark, PhD with high commendation, overall best thesis at UA for that year, that's 2016, that's the blue ribbon. Teresa Thompson Barnes, Trisha Miles Fraser, who is here in the audience, Gabrielle Andrade, Rochelle Davis, and those have stars there because for those, out of those research, we received the Principal's Award for Best Research Publication in that year. Or over time, right. And then I can't claim Tatrice Bachelor. I mean, she's from physics, but I was co-supervisor of that project, and Professor Tanisha would have mentioned that collaboration. And making cameo appearances, Rohan Grant, who is one of the ushers. Well, in fact, Chikwendo Onuoha is also an usher. So I will mention their work as well during the talk. So I have several affiliations and associations, and some of these have been mentioned. So most persons would associate me with WIGAT Jamaica. And pictured here, I'm the youngest of the stalwarts. So these were either present or past presidents of WIGAT across the university. And we were meeting in London for a pensions meeting. But the PhD was done at Cave Hill, and I spent some time at the Royal Tropical Institute in Amsterdam. KIT is Koninklijk Instituut voor de Tropen in Dutch, and I had to learn Dutch while I was there. International Leptospirosis Society. When I came back to Jamaica, I was at the forensic lab as chief forensic officer. Then I left there to go to Northern Caribbean University, and from there I got a fellowship to go to Loma Linda, registrar, where I did some work on oral microbiology. And we'll talk a little bit more about that a little later. American Society for Microbiology has seen quite a lot of me in terms of work that I have done. And the Pakistan Society for Microbiology, I am a mentor for many students and young faculty there in the subcontinent. International Society for Infectious Diseases, I was a council member, I have spent some time at the University of Minnesota, both School of Public Health and College of Veterinary Medicine. When there was the Fulbright Academy, because I was a Fulbright Fellow, and there are some that are here, some that are online, I was a part of the Academy, Royal Society for Tropical Medicine and Hygiene, the British Society for Antimicrobial Chemotherapy, the African Initiative Group for Microbiology, again, serving the Africa and the diaspora in terms of microbiology and the importance of getting things done there. And finally, the International Society for Antimicrobial Chemotherapy. So I've been around a little bit busy, trying to help as much as I can. But I started, and you see, VC principal, everybody claims you as your own. So, you know, you have to pay respect due. Wilbert Stewart Basic School was where I started, class of 74. Winard Road All-Age School then, now primary and junior high, class of 79, Arden High, class of 84, and then I went to sixth form, and then, of course, UA Mona, class of 89. But my love for microbiology really grew in my bachelor's program. So I did a double major. Back in the day, you could actually do a double major. And the poetry had significance. 70% for the final exam. It is a 3-hour exam, and you write and write and write for miles and miles and miles without stopping, hoping to get sufficient to pass the exam. The pass mark was 40%. And there were many students who were struggling to get the 40%. But in that time, the microbiology class was 6 persons. It was a level 2 course. So students were handpicked. Alan C. Weiss is to be blamed for that, but I survived and I graduated with upper second class honors, for those who are not aware. I disappointed quite a number of persons because I'd gotten an A in math. Actual science was calling me. Now I'm actually regretting it because it actually does pay a lot of money. Money answereth all things, right? So I worked on red peas and rhizobia with Professor Ahmad and Professor McLaughlin. And then I went on to do a PhD in microbiology with Professor Paul Levitt, where I studied postmortem leptospirosis and molecular epidemiology. That is how I looked then. So you see, all right, I have put this slide up really to show not so much my penmanship, which was decent, but, you know, the fact that, you know, you start with a clear aim and you have your objectives. And this was an EU-funded program. It was for 2 years, absolute. After the money runs out, you must figure out what it is that you're going to do. So I encourage my students, do not try this at home. 2 years for your PhD is difficult. But it is not only difficult because it is difficult. I'll tell you why it is difficult. So it was a collaborative effort between 2 countries in Europe, 2 in the Caribbean. So Royal Tropical Institute in the Netherlands, this is Dr. Rudy Hartskerl. Veterinary Services Division in Northern Ireland, this is the late Dr. Bill Ellis. Professor Paul Levitt, you would have mentioned that before, at the Leptospirosis Laboratory in Barbados. And then Carrick, it used to be Carrick, now CARFA, in Trinidad, and this is Dr. Franklin White. So I was based at the Medical Research Council Leptospirosis Laboratory. Then after they pulled out, then the Barbados government took it over. So that was where I was based, but attached to the Queen Elizabeth Hospital. There was no department of microbiology, so I was attached to the Department of Surgery and Pathology. And I was the first PhD from Cave Hill. And one of the interesting things about it, well, so my The chair was a psychiatrist, and you only did an oral exam if you were failing. But it was standard for a PhD to be examined by oral exam. There was no oral presentation by the candidate. It was muscle, 2.25 hours, I recall, of question and answer. And it was only ended because they had run out of questions. And I had an external examiner from the UK. I had Dr. Dorothy King from Jamaica, and I had my supervisor, and all of them were asking questions, no holds barred. But in the end, what I ended up doing was not so much what I had started with, but I had to change the objectives. And this is where now I had to become creative to now use molecular methods that could be used to detect and characterize leptospiral strains for epidemiological purposes. And I show my students, my first year graduate students, this slide, because everybody comes in to graduate studies thinking that success is a straight line. And you hit the bullseye, you get your degree, and you're home and dry. In fact, it is rarely the case. And many times you have to draw another bullseye after you have completed some circuitous pathway, and then you call it George, you get your degree, and then you move on. That is what happened. Because in my case, I would have had to do some things that were unethical if I was going to get the objectives that I had set out to do. Why leptospirosis? Well, it is considered a neglected tropical zoonosis, both emerging and re-emerging, depending on where you look. Outbreaks are possible in most cases, and in this map, you see that you can have outbreaks, and outbreaks have occurred anywhere. Anywhere where you have something that has a kidney, it can harbor lepto, it can be excreted in the environment, and persons can get it. And there is something that I want to show just a little bit. You have to know where to look. You have to be persistent. So it causes more than 1 million cases annually and 59,000 deaths. And the Americas are, bear the brunt of the burden of the disease. Clinical features, so there's protein, many clinical manifestations. It mimics several other viral illnesses and bacterial causes of what used to be called back in the day pyrexia of unknown origin. Means that somebody had a fever, they know what was causing it. Depending on the history, you might have to suspect lepto. Because if you don't, well, we're coming to that in a little while. So high degree of clinical suspicion and differential diagnosis because it mimics several others. So typhoid, encephalitis, dengue, HIV seroconversion, influenza, malaria, brucellosis, viral hepatitis, pneumonitis, hantavirus infections. Did I miss anything? No. So if you're not looking for it, you will not find it. And you will think that they have something else. And by the time you find it, it might be too late. Case fatality rate is about 10%. It can be as high as 30 or even 40%. And it might be biphasic. In fact, this is what the textbooks say. In most of the cases, patients come in when they are fulminant, icteric, have jaundice, and if they are not managed carefully, they will die. And as is the case many times. The incidence rate at the time in Barbados was 13 per 100,000. And it was, mainly among men, mild cases in children as compared to adults. But although we didn't experience that so much when we were there, females are more likely to die than males. Sounds like something else that we know. I'm going to skip through some of these, but acute renal failure, acute kidney injury is seen in up to about 40% of patients without any issues in terms of their urine output. That's what oliguria is. But if there is oliguria, then the odds of death increases 9-fold. If there's thrombocytopenia, which means that there is an issue with clotting of the blood, platelet counts are low, 50% of cases. That correlates with acute renal failure for poor prognosis. So again, so the organism is there, but if the patient is not managed, they could actually demise quite quickly. And there are some liver function tests, pancreatitis, things that can occur, which I will skip through. This slide is important for us to consider because for those who are involved in public health, they look at the disability-adjusted life years or the estimate of the burden of disease and the number of years that are lost because of the disease. Annually, 2.9 million dollars are due to lepto, which means that persons are sick and they take long to recover, and so productivity goes down, but also they cannot function because they're having, you think that COVID is more understandable, and COVID is a viral illness. Lepto looks like COVID in terms of long-term sequelae. So one of the questions that actually came up in my oral exam was why it is that persons with past lepto would suffer from headaches over several months, years. And this is a part of the reason. So it predominantly affects males. For comparison, it accounts for more than 70% of what we have for cholera. If you think cholera is bad, put that with lepto, put that with all of the other infectious diseases, and you realize that there are serious issues. That doesn't take into consideration the chronic non-communicable diseases. So it's actually quite significant. And of course, I mentioned about the preponderance of cases. In terms of the cost, rural areas globally bear the brunt of the cost for the disease. But in Jamaica, there was a paper that was published in 2018, which puts the cost, if you have to overnight in hospital, over $80,000. Because persons are coming in with severe illness and need to be, given several rounds of antibiotics, etc. Thing is that if antibiotics are not given early in the disease, it doesn't matter how much antibiotics you give the patient, it will not clear the organism, because the organism disappears from the blood, it finds itself in several organs. And so that causes a lot more challenges in terms of managing the disease. Historically, it was an occupational disease, and it was caused by various types of exposures. You look at the outbreaks that have occurred between the 1970s and 2012 is predominantly in the rural areas, that is the gray bar. But you could have any kind of exposure that could lead to infection. So these could be recreational exposure, living in slums, walking barefooted in damp conditions, because it was considered, for example, that the organism needed an abrasion to get into the body. In fact, if the skin is water-soaked, the organism will get in. And so anywhere you have flooding, there is a potential for infection. Gardening is an issue as well as rodent bites. Last week, there are two news items that I want to bring out. So this is in India. Last week, they're having a heat wave. They're having intermittent rains. And they're having cases of lepto. Prof. Tanisha, we had discussed when we were doing that paper that because of what was expected with climate change, that it might not be considered that lepto could be a significant force in the future. Well, that is put to rest, and other research that we have done will bear that out. But this also, that in fact, even if you have temperatures over 40 degrees, you can still have cases. But closer home, because the U.S. doesn't think that this is a problem in the U.S. In fact, just last week, there was a report that was coming out of New York City. They're blaming the rats. And, so 24 cases in the city in 2013, higher than in any previous year. The rats are invading New York. So we mentioned the diagnosis. So it can be diagnosed by dark field microscopy, which is unreliable because you cannot tell the difference between a lepto and a thread, a fibrin thread that is on the slide if it's not moving, because they look alike. You can use culture, but it is slow, but it can be done. You can use immunohistochemical or fluorescent staining that is definitive, and we have used it to identify leptos in the brain of humans. Serology is a mainstay, and serum is used for that, but it is notoriously difficult if you're using this particular test, the microscopic agglutination test. But ELISA has been used, and dipstick or lateral flow, which is similar to what you'd use for a rapid COVID test. A similar kind of test is applied, but they have some issues. And then, of course, we have molecular testing, which is PCR, which is fast becoming a mainstay. Some important things to note about lepto is that it is technically a gram-negative, which means that we use a particular stain in the lab. It should stain, well, it should not stain, but it should give you a pink color. In fact, it doesn't stain. It is one of those atypical organisms. And I have gram-negative in brackets because unlike other gram-negatives, it is sensitive to penicillin and doxycycline. It is unusual for you to have that. It can pass through filters, so it means that it can contaminate media that you're making up if you're using filters to filter. It can be grown in the lab, and the important thing about it for the microbiologists here is that it has 2 chromosomes. The second one is important. It is not a plasmid, and they use these things to good effect. All right, let me move quickly through these. But this now becomes one of the things that persons bear note of quite importantly. Most of the effects that are seen in terms of damage in the body is caused by the organism itself. It has a corkscrew motion. It causes damage by just boring through tissues, significant hemorrhage. So it will cause a similar kind of hemorrhage that you might see in dengue hemorrhagic fever, for example. But it also has a whole host of things that it can use to cause damage. It can kill cells. It can do lots of damage to anywhere where it finds itself. But one of the things that interested me was the fact that you could have damage even though the organism was absent. We saw this on some staining that we did while we were in Barbados. And it can induce autoimmunity, something again that we see in COVID. It took me 30 years. So this was 2003. This slide was made in 2023, and I had to present at an immunology conference last year, that this is how lepto can cause cytokine storms by causing dysregulation of the immune system. In a similar way that you would have for COVID, these are similar cytokines that you find in dengue, but these are produced later than when dengue would produce it. So you can imagine if somebody was infected with lepto and dengue, and the kinds of outcomes. So the kidneys can be involved after infection, the lungs, the brain, the heart, the blood clotting mechanism, the liver, the pancreas, so all of these multi-system. If everything collapses, then it's curtains for the patient. And one of the things that cause the problems is this balancing act between inflammation, which is the body's natural protection against invaders, and calming things down. So you have fought, now you must calm yourself and get back. If the person cannot manage that process, then severe infection will occur based on this model, and they could die. That is what the right side of the slide is actually talking about. All right, so some challenges that I had. I must talk about these, and I call this slide coming of age. 3 months hard labor at Royal Tropical Institute in Amsterdam. And we were looking at developing an internal control for the molecular testing that we're doing, because when you are looking at tissues and PCR, then you get a lot of negatives. Are these true negatives, or are they something that is inherent in the reaction? So developing this, we could spike the reactions and we could tell whether it was a true negative. Why this is important is that we presented this work in Italy in 1994. And there was a question that came up from, and this was a European conference, from somebody in the audience, a professor, who didn't think that this work could be done in the Caribbean. And for those kinds of questions, I leave my supervisor to answer. And he did deal with that matter, and it was put to rest. That group became the International Leptospirosis Society. I am more respected in the society now, thanks to that intervention. But another aspect, and this is now with collaborators, and 2 collaborators are here, and others that I've worked with. This is something else that I had to get over. So you have 2 papers that are here. One that I would have been the lead author on the top one. The second one is one that was published by our collaborators in Amsterdam. The bottom one is published in Journal of Clinical Microbiology. John, this was the top microbiology journal back in the day. I'm publishing it, sir. No, hold on. No. You get... We are submitting our work done in Barbados to J. Clin Microbiology. The editors tell us we can't publish there because they have a similar paper that is being considered for publication. So we had to go across the pond to J. Med Micro, which is number 2, and publish there. But you'll notice from the authors on the paper that they overlap significantly. These are our collaborators. Of course, the other aspect of it is that how can I be first author? Because students, John, do not become first author on international papers. For those who are asking for more first author publications, note that it is notoriously difficult if you're a collaborator. Well, this was the experience back in the day. Things are a little bit easier now, because perhaps principally it's because of the standing of UWI, but it was difficult to get a first author paper in those days. Okay. Molecular epidemiology just was born in that era, and we were a part of the pioneers using PCR to differentiate Leptospirosis species. So we had several that we had isolated, and we were able to identify them successfully, and we had good resolving power. 0.99, 0.96 were the high ones that we had there. The main part of the thesis, and I think I got about 3 or 4 papers from the work there, so not too bad. But we had 8 fatal cases. Medical students have to do 5, Monica, is that true? Still the case? Still the case? No? 8? 20? You are putting more work for the students to do? That's unconscionable. And they have to do a research project on that. That's wicked, Yeah, man, undergrad. Yeah, 5. Okay, all right. So 8 fatal, and then I had 5 control cases. All of these were males, 21 and 76. But importantly, all died within 6 days of admission to hospital. One of them just missed being a coroner's case because it was just outside the 24 hours. And these persons were waiting too long. In one case, somebody was turned back from the hospital, given some medications for pain and fever, and told to go back home. They came back in, and shortly afterwards they were in the morgue. It's sad. And, you know, and the values that you get when you do clinical chemistry, Dr. McGrowder, it can be normal, it can be high, it can be low. Lepto doesn't care, you know. And you can have tissue damage without, you know, tissue dysfunction without any physical damage. Because when we did the postmortem, the weight of the organ is normal, organ looks okay, but it has failed. And we were able to identify organisms by culture, from the eyes, that's the aqueous humor, from CSF, immunofluorescence from the liver and the brain, and from PCR from various organs. Important thing is that, like other spirochetes, Lepto will target the brain. In Lepto, there is no such thing as a blood-brain barrier. Now, antibiotics may not get in. So if the organism is already there, it might be too late. That's a challenge that we face when we come to Lepto. There was some time for service. So I was involved with HIV/AIDS work while I was in Barbados. And we offered home-based care for persons living with AIDS, assisted in persons who were in the hospital, and trained persons in churches. Again, back in the day, this was about 10 years after HIV was discovered. Persons were being turned out of churches, were being ostracized, not well taken care of in the hospitals as well. And so we were in a position that we could actually help, thanks to the UK government and the parent body. And we published a monograph in this book, AIDS Epidemic, Caribbean AIDS Epidemic. So when I came back to Jamaica, there was a hiring pause at USO. I went to the forensic lab and did DNA work with Mr. Beecher. And then after that, I went to NCU and went to Loma Linda. And one of the highlights for me in teaching molecular ecology or microbial ecology was projecting myself to be like one of the greats of microbiology. And why not? As I came to realize they were human beings just like me, just that they were probably a different color. But that has never stopped me before. I had the mantra, if it can be perceived and believed, it can be achieved. And I really went after what it is that I wanted. I was very fortunate to meet some important persons. Maybe Prof. Watson is not that important anymore. But anyway, he got the Nobel Prize for discovery of the structure of DNA. So this was 50 years after the discovery. We met at a conference in Miami. I also met Prof. Barry Marshall, who confirmed that gastric ulcers were caused by a bacterium and not by stress. Yes, Mommy Rose. It's an organism so treatable with antibiotics and steroids. And then I met Dr. Kathleen Kate Rubins, who at the time was just coming back from the International Space Station. And I still have some lofty dream that space may be the final frontier. You know the date, VC. This is May 31, 2017. This is also significant because I think that was the formal announcement for the professorial. So I was on the council for ASM at the time, and I was there with 95 other members, one other black person in an international society. That was something to talk about. But because my brother had died a few days after that, then everything was shelved in terms of doing any kind of celebration. In fact, 2017 was one of those years that, well, you have to remember it, but, you know, that was how it was. But in considering how you're going to proceed in a career in microbiology, especially working on Lepto, you have to understand where we are in Jamaica as far as the other places where the disease occurs. If you look at the slide, you'll see that Jamaica is well down the wicket in terms of the annual incidence rate. And even when you come a little bit closer, we are nowhere near India, Philippines, Brazil, Thailand in terms of case numbers. So how is it that I'm going to actually make a name among areas that have much more diseases? So it means that I need to be creative. I need to be doing something that they are not doing, and I need to champion that quickly. Okay. The only thing that I'll mention in this slide is that rats get a bad rap. If we kill all the rats, we will still have lepto, is the purpose of this slide. And one of the important things, VC, is that whistling frogs, Barbados, and toads actually are important in the scheme of things in Barbados. And for those, if you get a very small drop of blood from a whistling frog, those are the ones that make those noises. Right, okay, right. The more you know, perhaps the more you have to fear, but it's not the idea of this lecture. So lepto naming follows the Salmonella protocol. So you have genus, species, then you have a serovar, and then a strain. I mean, that is how the naming goes. So really just highlighting this for persons who might be so disposed. As far as Jamaica is concerned, it was first identified in 1953. And I usually get some guesses when I ask people where would Portland Veer come from? Portland Cottage and Veer, so that is Clarendon. Because, some persons think that Portland is in the east and so, and Moneymusk, St. Catherine, Bogveer is out. So there are some strains or some serovars that are endemic to Jamaica, not found anywhere else. And then there is Jules, which was originally from Zaire or Republic of Congo, which is only found in Jamaica now. So we have our own, which will cause us no ends of problems. So rats, mongooses, house mice, and humans. Yes, all the ladies that are afraid of house mice and think they are not your friends. And we have, unfortunately, found some serological evidence for bats carrying lepto as well. I don't think there are many vets that are in the house, but perhaps there are some of them that are online. And we have quite a lot of carriage in our livestock in Jamaica as well. So as far as our clinical postmortem cases in Jamaica, serovar Jules and Portland Veer and Icterod, those cause the majority of cases. 3.4 males to 1 females in terms of the risk profile, and all ages are involved. It is highly linked to climate, so only in the second rainy season. And so we have about a 90% correlation with the second rainy season. We had an NHF study that was done which looked at fatal cases in Jamaica, and we found organisms by PCR in the heart, lung, eyes, among other places. So it means that the organism is actually causing significant sequelae locally as well. All right, so we mentioned that there is climate variables, and so this was a work done with the Department of Physics. So it is tied to El Nino. If La Nina is here, it means that we get more cases earlier, because there's earlier rainfall in this period that we call the dry season. So it is actually not looking good. All right, so there were some links that we had with the University of Minnesota. And the bacteria probably were actually laughing at me because now they got me we have always kept them at -20, -4 Fahrenheit. And this was a high temperature in January when I was there to work with some collaborations there. And I was actually getting over that hurdle that I had. This is Professor Johnson of EMJH. So I met him. He was retiring. In fact, I was being offered a job at the time, Vice Chancellor, and I was so committed on my J visa that I didn't take up the offer. And maybe because it was too cold at the time I said. I went back on a Fulbright fellowship in the summer, better time to go. And clearly I was not in the lab all the time, so we were out fishing. And then I went back again for a sabbatical in 2013. And this caught me grilling, doing jerk chicken on the grill, just before winter started. In fact, the day after this, now this is September, and snow started the day after that. It reminded me why I said that I would not. And not because of George Floyd, but, But it was during this time that I learned about Moodle and how to actually conduct online courses using the platform. In fact, they were using, 2.5? At the time, we were using 2.4, so I was a little bit ahead of where Mona was in terms of. So once the pandemic came, I had some working knowledge of how Moodle worked to the chagrin of the students, because I was actually preventing them from doing things, because you can actually control the students' time and do quite a lot of things as far as your teaching is concerned. Water is important for Lepto. We did a study across surface waters locally. It was a snapshot study. We didn't find any organisms. We found Giardia, John, in one location, which will remain nameless. But we know that once there is runoff from farms or other places, then that water can become infectious, and that has actually happened. But in households that had cases, water that is harvested can be something that is infectious. So we found a small number of water samples, that persons that caught up runoff from the roof, etc., that had organisms in there. And these actually implicated Portland Vera and Ictero, which are the ones that are found more in the environment. And important, I wanted one or other of the ministers to be here, but the word will get back. Rural communities, presence of a garbage dump, and of course, organisms in the water correlated with households with a history of leptospirosis. Very important, but it seems to be more a strong case for those persons who are in rural communities as compared to urban communities. Let's say something about rats that we have in New Kingston, for example, there is another version of that story about the country rat and the town rat. And it is centered in the story of leptospirosis. Some work that we did with butchers and their associates. We have problems in Jamaica. We have some serious problems. It has to do with how persons use personal protective equipment. So many of these had them hung up, and they were in the carcasses with their bare hands, bare feet, and these were meat that were going to be for consumption. The challenge is that some of these animals were actually positive for lepto. Unhealthy behaviors, younger age, lower education, limited knowledge base about the disease, stray dogs and rodents, and maintenance issues were some of the challenges that they faced at the time when we did the study. I won't ask anybody who may know what the situation is now. Perhaps we don't want to know. Things have improved? Oh, God. Another aspect of the disease is that if you don't look for it, you may miss it. So how most diagnosis occurs is that if there's a sample taken, for example, for dengue, it is not going to be investigated for lepto or anything else. If they don't find dengue, they didn't have dengue. So with John Lindo and some other collaborators, we look back at one of our more significant outbreaks that we had in Jamaica. And we examined those cases, about 3,500 or so of them out of the 4,000 odd. And we found, expectedly, a high proportion of them with dengue, but also that about 6% of them were positive for lepto, meaning that they were having an active case. Because we did not want to cause any alarm with our collaborators elsewhere or with external partners from the USA, we look for IgG for malaria because we did not want to identify any active case. So IgG indicates that there was previous exposure, and we found 6.5%. Sometimes what you don't know can harm you. And that is a very important aspect as it relates to microbiology. What doesn't kill you? Stress. So some work from Tricia. We found that temperature oxidative stress actually can be triggers for virulence, how it is that organisms will cause disease. And effectively, without getting into any of the details, the organisms are already primed with higher temperatures to cause problems. It doesn't matter where the organism will come from, they are on high alert for disease causation, irrespective of where they come from. But we found that those that are more environmental associated are more influenced by the variations in climate. So we say bigger is better if you want to do road. So it means that you have more opportunities to cause disease, etc. And then, of course, there was another aspect that we found was that Leptospira is able to make its own attachment even if you remove the usual means for it to attach to cells. Now, what do I mean by that? For an organism to cause disease, it must attach to a surface, and it uses various receptors that are on this surface. We use enzymes to cut those off, and the organism was still attaching, albeit lower. So it means that it is able to build its own scaffold to effect binding and infection. It means that there is, it's hard to deal with an organism that is actually moving ahead. I like this picture. It means that I'm transitioning away from lepto. And I will use one or two examples from this and then I close. I think we have heard quite a bit. So the prevailing theory of microbiology and infection is that all organisms they sing on the choir. They are naive, they come with a halo, they will cause no problems, they are our best friends. And then something happens. And then they can either become antibiotic resistant or they can become virulent and cause disease. And there are several reasons that we know for this. They can find genes, take it up. They can have a mutation and they change. Or they can join with another organism, sexual activity, conjugation. Stress is an important aspect, and they will actually increase the number of changes that occur at a time. And we see this with COVID. And there is co-evolution. So it means that as the body tries to mount a defense, they are mounting an offense to counteract that. We call that the Red Queen effect. That's co-evolution. Perhaps the most significant one here is the effect of viruses. So viruses are like mosquitoes to us. So if you want to compare, a mosquito to us is annoying, can cause problems and disease. A virus to a bacterium can cause problems. But it can also be helpful in terms of transferring disease. But one of the important things that occur is that there is CRISPR-Cas system that is in place. I won't tell you about it, but it is really a system that the organism, the bacterium, has to prevent viral infection. Like a scissors that will chop up the gene that comes in. How is that important? We have taken it, medical science now, and that now is the basis of gene editing. So we can use the same kind of system to edit any gene in the human body. And I think Professor Doudna got a Nobel Prize for that in 2020. The challenge with these things is that the technology advances well ahead of regulation, because scientists are thinking, and they are thinking outside of the box, and they are not afraid to, to test and extend, the boundaries that are possible. So it calls for concerted action as far as how it is that we're going to manage. Okay. The other thing that we're seeing is that the organism doesn't have to choose one or other path. They actually can choose both. More genes mean that they need more time to replicate that, because it's a bigger thing that you have to multiply. In fact, I tell my students, microorganisms don't read microbiology textbooks. So they don't care what we think about them. So instead of taking longer to replicate, they're actually doing it faster, even though they have more things that they need to replicate. So as I said, just a couple of examples that I'm going to speed through. There's a lot that is said about what is in our chicken, what is in our food, etc. And the manufacturers might not be telling a lie. It's just that if it is found, they don't know where it is coming from. So we found, for example, antibiotic-resistant E. coli from day-old, I think, to about 42-day-old chickens, which for 6 months, the farmers did not have any evidence of antibiotics being used on the farm. 6 months. Day-old to 42-day chickens, antibiotic-resistant organisms. Where are these coming from? John, that's a question for you. You have no idea. A lot of it might be coming from the environment or coming from animal feed. We need to confirm that. But we know that a part of what goes into animal feed is from antibiotic manufacture, all of those spent things that come. We also found, and maybe I'll use this as the second to last item and then go to the end. I don't want to have you here past 8 o'clock. Very important thing, and of course, this is urinary tract infection, affects mainly women. We thought that it was important just to check what is happening, and lo and behold, the organisms were actually making a switch from one mechanism of resistance, that is sulfamethoxazole trimethoprim, which was the standard for urinary tract infection at the time. And they were incorporating something that was genetically passed from one organism to another on what we call plasmids, to be resistant to fluoroquinolones. These are second, third generation. Some of these are synthetic antibiotics. So these are not caused by mutations that will cause them to become resistant. These were promiscuous organisms that were sharing. What we found was that they were not just using one type, up to 4 types of genes that they were combining for resistance. And we noted this, and of course, as a result, Dr. Stacy went on to actually do some characterization. These were housed on some large gene cassettes that were mobilized between organisms. And they had a mechanism for insertion in another genome to cause the problems. Last thing that I will say, and then I go to the end. There are some issues that we see with organisms. So the organism does not need to have a gene for antimicrobial resistance to survive in the presence of antibiotics. So if they have something that they can cause somebody else who is in their vicinity to do the work for them, then they will gladly oblige. So in this case, there was a pigmented organism which is producing pyaverdin, which is an iron chelator. So it grabs all of the iron in the environment. It prevents the other organism from growing well. But because it doesn't want to die, it will release some of the iron. The antibiotic-resistant organism can produce sufficient of what is needed to detoxify the antibiotics. Both of them survive. This is called the black queen hypothesis. It's what organisms do. They cheat. Principal, because you are in that area, this is also called the microbial consumer resource model. So by managing what is there, then it serves another good. So it becomes public goods. Because you have it, what you have is mine, and so I will cause you to do what I need you to do, and we both benefit. But I'm going to get the lion's share of the benefit. We're learning a lot of things from microbes, and this is how humans work as well. All right, I'm going to skip to the end in the interest of time. Okay, so what we have been talking about this evening is that both nature, that is genes, and nurture, the environment, influence antimicrobial resistance, they influence fitness, that's the ability to multiply, and they influence the ability to cause, disease, and all of these together will influence the outcome or the prognosis, whether the patient will live or die. Genes can be lost over time, as seen in Lepto. They can be influenced by culture or peer pressure. They can be borrowed via cooperation. They can be traded or shared, or they can be handed down. And they come in all sizes. Some policy implications. So greater understanding of pathogens, antimicrobial resistance, and virulence genes, and the role of external drivers hopefully will lead to greater focus on diagnostics, vaccination, and therapeutics. And there is need for clearly a greater emphasis on One Health, because it's all of us that are together as far as the environment is concerned. And if we don't take care of the animals, well, the next pandemic is likely to be like the one that we're just coming through, which is a zoonotic infection. And we need to look at the multiple determinants of health. Right, Dr. Cunningham-Myrie? Including the political determinant of health, because there are some aspects that require political will if it is actually going to get done. Climate change and increase in temperature and flooding worldwide will likely cause a significant increase on the human, veterinary, and economic costs of infectious diseases. So clearly, there need to be greater focus on surveillance and mitigation effects. But we're reminded that it is a microbe that will have the last word. Because while we are planning, they are wiping. to use, to corrupt a particular phrase, registrar. There's some other work that we have been involved in, and Rohan and I, we have looked at gut microbiome in health and disease, and we looked at 42 healthy undergrad students. We found that there was no difference, and to the dismay of the Trini posse, which was a part of the team, doesn't matter if you're a Trini or Jamaican, the organisms are similar. We have recently received a D43 grant, and this is a UWI-SUNY collaboration looking at periodontitis and associated systemic comorbidities. And we have been interested for some time on mixed species biofilm cultures. Are these interactions antagonistic, mutualistic, or synergistic? From these data, if you grow them in culture, they don't do well together. So in fact, staph will kill Pseudomonas in these cultures. But when there is a threat, like heavy metals, which is important in microbiology, silver and other things are used in the hospital setting, they will collaborate and become tolerant to those agents. Disinfectants, at the time when we were doing this study, there were recalls of a 4 times concentrated disinfectant that is used in many homes. Interestingly, Jamaica didn't have a recall of those products. It was pine sol, was it? 4 times concentrated. Barbados had a recall, but Jamaica didn't. So we're finding that organisms together, the power of 2 or 3 registrar, can actually resist what would normally kill an organism. And we are interested, principal, and we need to get one on board, a scanning transmission electron microscope, because there are quite a number of work that we need to do. So I had a student who was looking at biofilms in the food industry, and she did her work in Canada. And for that, unfortunately, because of the nature of the work, both examiners were external. So we could not find a suitable internal examiner for that student because of the nature of that work, because nobody was really doing that kind of work locally. And this was in the Canadian food industry that she actually did the work. And finally, metagenomics and climate, looking at, well, what is in our wastewater? So it is actually a very good indicator, a sentinel of the health of a population. It has been used extensively in COVID. So in fact, it can tell you what is happening well before you get symptoms of persons being ill. We have access to treatment plants. It is something that I have activated, and there is some collaboration that I'm working on with the University of Illinois. Urbana-Champaign, is it? Yes, and so we have started talking about developing a project. Finally, registrar, finally. Lessons. You can succeed even in the most trying or stressful circumstances. When faced with a common threat, even sworn enemies will collaborate and cooperate. Don't listen to the naysayers. It can be done. It might just take longer, but it can be done. Occupy niches. And Marvin is not here either because I would rag him as well. In other words, working in silos can be good for your health. You have learned this from the microbes. Yes, I know teamwork is good, but for your health. But very importantly, and I'm not knocking teamwork, but in teamwork, it means different things to different members of the team. There are some that are going to be directing, like I know Patricia will do very effectively. I'm starting to rag persons already. And you always have to be working with these. So it is nice to say, yeah, man, teamwork makes the dream work. Yeah, right. Until you start to work with some people who will not pull their weight and expect you to do all of the things. And registrar, I think this one is for you. This last one, number 7. perhaps God also listens to microbial prayers. Because if it wasn't the case, then every time that we say grace and we pray that God cramp and paralyze, then nobody will get a foodborne illness. Nobody would get sick. But then we are seeing different things. Thank you very much. It has been my pleasure. So you do everything at least once, so a 5K. At least once, I did. Right, Registrar, over to you. You want to switch to the... You could if you wish. It's up to you.
Please give the professor another round of applause. Although it is not on the program, but I think it is appropriate to entertain one or two questions. I don't know if somebody's online who would know, but from in the audience here, are there any questions, or Professor?
Everybody has been mesmerized and beaten into subjection. All right.
Well, I think it was all clear. I mean, if I survive 2 hours of a lecture and don't sleep, it must be interesting and very clear. So thank you very much, Professor. Please give him another round of applause. And I'm sure you'll agree that this has been a very interesting journey for Professor Brown, and very interesting findings and research findings, etc., that have implication for policy, etc. Prof. Brown, you know, I'm a son of Clarendon, right? From Vere. So I'm happy that where I come from made a contribution to your to your career in terms of the Portland Vere and the Bog Vere and the Money Musk. That's where I come from, and you know, strong people come from there. I say I survive without having leptospirosis. So, ladies and gentlemen, it is left for me to thank you. I want to thank Vice Chancellor and the leadership of the university, who have shown serious support to the scholarship of Professor Brown, coming here and supporting him this evening. Please give a round of applause. I want to thank the family members of Professor Brown, obviously, who have been supporting him right through. His wife and children and in-laws. Please give them a round of applause. We want to thank colleagues, not only from the Faculty of Medical Sciences, but the cousins from the Faculty of Science and Technology. Give them a round of applause. It's a wonderful testimony to collaboration and you know, and all of us who have come this evening. I want to particularly thank Guardian Life, who have sponsored the refreshment that we will partake of in a little while. We want to thank those who are online, who have, you know, I suspect more people would be online, according to Professor Brown, from all over the world. And since it's on YouTube, I suspect that in time to come, more and more people will view it. I want to thank our saxophonist. Give him a round of applause again. I want to thank those who bring greetings, Principal, Professor Stevenson, and Professor Gus Williams. Please give them a round of applause. And in his absence, our Deputy Principal, Professor Marvin Reed, who gave the the introduction. And we thank our colleagues from University of Buffalo who are here to show solidarity with Prof. Brown. Please give a round of applause. So ladies and gentlemen, this brings the curtain down to another in our series of professorial lectures. And we have a very busy space here, so I'm sure very soon we'll invite you to another one because, as I said, we are just flowing with these things and we want to keep the momentum up. Thank you so much. God bless you. Thank you, Hilda, for your prayers. You know, give the Hilda a hand, please. Thank you very much, ladies and gentlemen. Please enjoy the refreshments. God bless you all.